Welcome back to Cancer as a Mitochondrial Metabolic Disease Basics playlist. What if cancer isn't just a disease of random genetic mutations, but a disease of mitochondrial dysfunction? Many studies suggest that changes in mitochondrial number, structure, and function may be one of the earliest and main drivers in cancer. Every cell in your body depends on mitochondria to produce energy. In cancer, dysfunctional mitochondria can explain everything from abnormal metabolism to the uncontrolled growth seen in cancer. In this first video, we'll look at what the scientific literature actually says about mitochondrial dysfunction in cancer, review some basic cell and mitochondrial anatomy, and introduce the different types of dysfunction that may be involved. So without further ado, let's get into it. This is such an important topic to cover on a basics playlist. It basically sets the tone and the understanding for everything that is proposed for ketogenic metabolic therapy. And so we have to look at this very systematically, and it helps us understand what to do going forward, and everything makes sense. So before I get into the nitty-gritty, let's just take a look at the relationship between mitochondrial dysfunction and cancer. And when I just type that into the National Library of Medicine, PubMed, we're going to see that there are 17,000 plus results. And you're going to also see that that has been ramping up slowly over the past maybe 5 to 10 years because people are starting to really realize that there is something to this. And so I just wanted to show you guys this first because it's showing how and where medicine is going. It's going away from the genes and it's going towards mitochondria. And I think that this is kind of an exciting time because hopefully this innovation and this understanding will help us create solutions for the problem and not just keep pounding the same solutions into you that don't necessarily get to the root cause of the issue. So let's do a quick anatomy lesson of a eukaryotic cell, which is what we all are. And just like the body has organs, the cell has organs also, but they are small, so they call them organelles. And so here's the nucleus, for example. Here's where the DNA is at, the main amount of DNA is at. And then here, this little oblong shaped here is the mitochondria, and we're going to zoom in on that. So imagining we're zooming in, and we see here that there's a little more detail in this particular picture. So first of all, we have an outer membrane, we have an inner membrane, and we have a space in here. And these little in-pouching would be considered cristae. And then these little circular items are kind of zoomed in again. And then we're also going to see that mitochondria have their own DNA. Now let's take that and let's zoom in even further. But instead of doing 3-dimensional, we're going to do 2-dimensional to really get a sense of where things are. So here's OMM, outer mitochondrial membrane. Here's IMM, inner mitochondrial membrane. Here's a cristae, like I mentioned. This is the inner membrane space. Inner membrane space is here. The TCA cycle, which we've gone over, is in the middle here. And then here's the electron transport chain, or the OXPHOS system here. And then here's mitochondrial DNA. And there's a lot of places where this organelle can go wrong. And I'm going to pull this paper that was published in 2013 December. It's called Mitochondrial Dysfunction in Cancer. And it says here, a mechanistic understanding of how mitochondrial dysfunction contributes to cell growth and tumor genesis is emerging beyond Warburg as an area of research that is underexplored in terms of significance for clinical management of cancer. That's the understatement of the century. Work discussed in the review focuses less on the Warburg effect and more on mitochondria and how dysfunctional mitochondria modulate cell cycle, gene expression, metabolism, cell viability, and other established aspects of cell growth and stress responses. There is increasing evidence that key oncogenes and tumor suppressors modulate mitochondrial dynamics through important signaling pathways, and that mitochondrial mass and function vary between tumors and individuals. But the significance of these events for cancer are not fully appreciated. We explore the interplay between key molecules involved in mitochondrial fission and fusion and apoptosis, as well as mitophagy, biogenesis, and spatial dynamics of mitochondria, and consider how these distinct mechanisms are coordinated in response to physiological stresses, such as hypoxia and nutrient deprivation. Importantly, we examine how deregulation of these processes in cancer has knock-on effects for cell proliferation and growth. We define major forms of mitochondrial dysfunction and address the extent to which the function consequences as such dysfunction can be determined and exploited for cancer diagnosis and treatment. And we're going to look at this from a very basic perspective. We're not going to get into every one of these little things, but I just want to go over this. So for example, the membranes can be depolarized or not have a membrane potential, which means there's no electrical chemical gradient that are made by the electron transport chain, which is what's responsible for making energy and ATP. There could be hyperpermeability of the membranes where ions like calcium could be either accumulated, and that accumulated calcium could swell the mitochondria and lead to less efficiency. There could be defective respiration, which we're going to focus a lot on when we're talking about mitochondrial DNA mutations. So these two are very interrelated. And then there's misfolded proteins. I would also put in this dysfunctional mitophagy or quality control mechanisms. And then there are defective iron sulfur clusters, biosynthesis. And then there's either excessive or not enough apoptosis, which can maybe make a cell that would normally kill itself because there's major problems not kill itself, and those damages accumulate and kind of snowball out of control where you have a cancer situation. And this is another graphic that's part of this paper. And so here's like some hints of where we're kind of going with this particular video is we're going to be looking at mitochondrial metabolites and how they change gene expression, how mitochondrial ROS production can change signaling, but also can change mitochondrial DNA and further kind of snowball things. And then there are signaling issues. And then there are also quality control and calcium homeostasis issues as well. So I think that without getting too deep into these problems as a whole and kind of unpacking what a problem with hexokinase 2 or ABL or AMP kinase, that would be too much for this video. But I'm just trying to highlight that this organelle, which is so critical for energy production and many, many other things, there's a lot of places where this can go wrong. And one of the places where this can go wrong that's not really highlighted in that last paper is the changes in mitophagy. Mitophagy is mitochondrial specific autophagy, and that's the recycling of either entire cells or cellular components. In this particular subject, we're looking at recycling of damaged mitochondria. So when mitochondria are damaged beyond a certain point, they can go through a quality control mechanism. And if those quality control mechanisms fail, then they can just recycle the entire mitochondria. So that way there's no downstream problems that can function. So if you have abnormal mitophagy or abnormal recycling of mitochondria, that can lead to abnormal cells and cancer creation. That's another way that mitochondrial dysfunction can lead to cancer. The mitochondrial DNA itself is critical because the mitochondrial DNA itself encodes for OXPHOS proteins. That's what these color-coded annotations are signifying. So here's a pink region. Here's COX-1, 2, and 3. Or cytochrome B, which is this right here. Or this part of cytochrome 1, which is of complex 1, which is encoded in this red area here. So my point is that when there are DNA mutations within the mitochondrial DNA, we're not talking about the nuclear DNA, we're talking about the mitochondrial DNA. Whenever you damage this mitochondrial DNA, that has the ability to damage the encoded proteins on the OXPHOS system, and that can lead to problems also. That's highlighted pretty well right here. Here's August 2020, significance of mitochondrial dysfunction in cancer. It says here in the abstract, as an essential organelle in nucleated eukaryotic cells, that's all of us, mitochondria play a central role in energy metabolism, maintenance of redox balance, and regulation of apoptosis. Mitochondrial dysfunction, either due to TCA cycle enzyme defects, mitochondrial DNA genetic mutations, defective mitochondrial electron transport chain, oxidative stress, or aberrant oncogene and tumor suppressor signaling, has been observed in a wide spectrum of human cancers. In this review, we summarize the mitochondrial dysfunction induced by all these alterations that promote human cancers. And I picked this graphic because it so beautifully illustrates how this is all interrelated. So when we have damage of mitochondrial DNA due to oxidative stress and mutations, that's going to lead to problems with the proteins that DNA encodes for. And since those proteins encode for these mitochondrial electron transport chain proteins, that's going to lead to this mitochondrial electron transport chain being damaged in some way or defective in some way, and generally more leaky of reactive oxygen species. That's going to lead to oxidative stress, and it's going to ultimately set us up for cancer downstream. We're going to look at this in greater detail in probably video 3 or 4 of this mitochondrial dysfunction series for cancer. The other way that mitochondrial dysfunction can lead to cancer is through something called oncometabolites. And we're going to unpack that pretty strongly in the next video, as a matter of fact. But basically, what I want to do is give you a little bit of a basic overview. When some of these TCA intermediates, such as fumarate or succinate or malate or citrate or whatnot, build up, they can have changes to the nuclear DNA and cause epigenetic changes that can lead to cancerous kind of transformation. And then other ways that it can happen is, as it says here, succinate can lead to HIF1 stabilization, or fumarate can lead to HIF1 stabilization. That's called a pseudohypoxic stimulus. And HIF1, which we have not really gone over too much in this basic playlist, is a major player in the malignant transformation of cancer and the metabolic reprogramming that happens. So this is another way that mitochondrial dysfunction can set up cancer and sustain cancer through all of these mechanisms. So again, we can have mitochondrial dysfunction and energy production issues through a variety of ways, whether it be membrane depolarization, permeability issues and swelling of mitochondria, defective respiration related to defective mitochondrial DNA, defective quality control, which leads mutations and other problems to build up, and problems with not signaling for programmed cell death or apoptosis when needed. And then again, quality control, part of that is mitophagy, or getting rid of damaged mitochondria. If we don't do that, that means that these mutations and problems can build up, which can lead to cancer. Those mutations can directly affect OXPHOS performance and energy production, which can lead to cancer. And then lastly, when we have either enzyme problems, deficiencies, or toxicities, then we can have these toxic intermediates, or these TCA intermediates build up, which can then stabilize HIF and lead to metabolic reprogramming. So there's a lot of different ways, as this is kind of an overview of how mitochondrial dysfunction can lead to cancer. And that's why this research is ramping up, and there's 17,000 plus results when we search for mitochondrial dysfunction in cancer, because there are a lot of ways that mitochondrial dysfunction can not only set you up for cancer, but also sustain cancer once you actually have a tumor present. So if you like videos like this, please like, share, and subscribe. And until next time.